Chapter 9. How Cells Harvest Energy. Concept Outline. Section 9.1. Cells harvest the energy in chemical bonds. Using chemical energy to drive metabolism. The energy in CH, CO, and other chemical bonds can be captured and used to fuel the synthesis of ATP. Section 9.2. Cellular respiration oxidizes food molecules. An overview of glucose catabolism. The chemical energy in sugar is harvested by both substrate-level phosphorylation and by aerobic respiration. Stage 1, Glycolysis. The 10 reactions of glycolysis capture energy from glucose by reshuffling the bonds. Stage 2, the oxidation of pyruvate. Pyruvate, the product of glycolysis, is oxidized to acetyl-CoA. Stage 3, the Krebs cycle. In a series of reactions, electrons are stripped from acetyl-CoA. Harvesting energy by extracting electrons. The respiration of glucose is a series of oxidation-reduction reactions which involve stripping electrons from glucose and using the energy of these electrons to power the synthesis of ATP. Stage 4, the electron transport chain. The electrons harvested from glucose pass through a chain of membrane proteins that use the energy to pump protons, driving the synthesis of ATP. Summarizing aerobic respiration. The oxidation of glucose by aerobic respiration in eukaryotes produces up to 3 dozen ATP molecules, over half the energy in the chemical bonds of glucose. Regulating aerobic respiration. High levels of ATP tend to shut down cellular respiration by feedback inhibiting key reactions. Section 9.3 Catabolism of proteins and fats can yield considerable energy. Glucose is not the only source of energy. Proteins and fats are dismantled and the products fed into cellular respiration. Section 9.4. Cells can metabolize food without oxygen. Fermentation. Fermentation allows continued metabolism in the absence of oxygen by donating the electrons harvested in glycolysis to organic molecules. Life is driven by energy. All the activities organisms carry out, the swimming of bacteria, the purring of a cat, your reading of these words, use energy. In this chapter, we will discuss the processes all cells use to derive chemical energy from organic molecules and to convert that energy to ATP. We will consider photosynthesis, which uses light energy rather than chemical energy, in detail in Chapter 10. We examine the conversion of chemical energy to ATP first because all organisms, both photosynthesizers and the organisms that feed on them, like the field mice in Figure 9.1, are capable of harvesting energy from chemical bonds. As you will see, though, this process and photosynthesis have much in common. Section 9.1. Cells harvest the energy in chemical bonds. Using chemical energy to drive metabolism. Plants, algae, and some bacteria harvest the energy of sunlight through photosynthesis, converting radiant energy into chemical energy. These organisms, along with a few others that use chemical energy in a similar way, are called autotrophs, self-feeders. All other organisms live on the energy autotrophs produce and are called heterotrophs, fed by others. At least 95% of the kinds of organisms on Earth, all animals and fungi, and most protists and bacteria, are heterotrophs. Where is the chemical energy in food, and how do heterotrophs harvest it to carry out the many tasks of living? Figure 9.2. Most foods contain a variety of carbohydrates, proteins, and fats, all rich in energy-laden chemical bonds. Carbohydrates and fats, for example, possess many carbon-hydrogen, CH, as well as carbon-oxygen, CO, bonds. The job of extracting energy from this complex organic mixture is tackled in stages. First, enzymes break the large molecules down into smaller ones, a process called digestion. Then, other enzymes dismantle these fragments a little at a time, harvesting energy from C, H and other chemical bonds at each stage. This process is called catabolism. While you obtain energy from many of the constituents of food, it is traditional to focus first on the catabolism of carbohydrates. We will follow the 6-carbon sugar, glucose, C6H12O6, as its chemical bonds are progressively harvested for energy. Later, we will come back and examine the catabolism of proteins and fats. Cellular respiration. The energy in a chemical bond can be visualized as potential energy borne by the electrons that make up the covalent bond. Cells harvest this energy by putting the electrons to work, often to produce ATP, the energy currency of the cell. Afterward, the energy-depleted electron, associated with a proton as a hydrogen atom, is donated to some other molecule. When oxygen gas, O2, accepts the hydrogen atom, water forms, and the process is called aerobic respiration. When an inorganic molecule other than oxygen accepts the hydrogen, the process is called anaerobic respiration. When an organic molecule accepts the hydrogen atom, the process is called fermentation. Chemically, there is little difference between the catabolism of carbohydrates in a cell and the burning of wood in a fireplace. In both instances, the reactants are carbohydrates and oxygen, and the products are carbon dioxide, water, and energy. C6H12O6 plus 6O26CO2 plus 6H2O plus energy, heat or ATP. The change in free energy in this reaction is minus 720 kilocalories, minus 3012 kilojoules, per mole of glucose under the conditions found within a cell, the traditional value of minus 686 kilocalories, or minus 2870 kJ, per mole refers to standard conditions, room temperature, 1 atmosphere of pressure, etc. This change in free energy results largely from the breaking of the 6C-H bonds in the glucose molecule. The negative sign indicates that the products possess less free energy than the reactants. The same amount of energy is released whether glucose is catabolized or burned, but when it is burned most of the energy is released as heat. This heat cannot be used to perform work in cells. The key to a cell's ability to harvest useful energy from the catabolism of food molecules such as glucose is its conversion of a portion of the energy into a more useful form. Cells do this by using some of the energy to drive the production of ATP, a molecule that can power cellular activities. The ATP molecule. Adenosine triphosphate, ATP, is the energy currency of the cell, the molecule that transfers the energy captured during respiration to the many sites that use energy in the cell. How is ATP able to transfer energy so readily? Recall from Chapter 8 that ATP is composed of a sugar, ribose, bound to an organic base, adenine, and a chain of 3 phosphate groups. As shown in Figure 9.3, each phosphate group is negatively charged. Because like charges repel each other, the linked phosphate groups push against the bond that holds them together. Like a cocked mousetrap, the linked phosphates store the energy of their electrostatic repulsion. Transferring a phosphate group to another molecule relaxes the electrostatic spring of ATP, at the same time cocking the spring of the molecule that is phosphorylated. This molecule can then use the energy to undergo some change that requires work. How cells use ATP? Cells use ATP to do most of those activities that require work. One of the most obvious is movement. Some bacteria swim about, propelling themselves through the water by rapidly spinning a long, tail-like flagellum, much as a ship moves by spinning a propeller. During your development as an embryo, many of your cells moved about, crawling over one another to reach new positions. Movement also occurs within cells. Tiny fibers within muscle cells pull against one another when muscles contract. Mitochondria pass a meter or more along the narrow nerve cells that connect your feet with your spine. Chromosomes are pulled by microtubules during cell division. All of these movements by cells require the expenditure of ATP energy. A second major way cells use ATP is to drive endergonic reactions. Many of the synthetic activities of the cell are endergonic, because building molecules takes energy. The chemical bonds of the products of these reactions contain more energy, or are more organized, than the reactants. The reaction can't proceed until that extra energy is supplied to the reaction. It is ATP that provides this needed energy. How ATP drives endergonic reactions. How does ATP drive an endergonic reaction? The enzyme that catalyzes the endergonic reaction has two binding sites on its surface, one for the reactant and another for ATP. The ATP site splits the ATP molecule, liberating over 7 kilocalories, 30 kJ, of chemical energy. This energy pushes the reactant at the second site uphill, driving the endergonic reaction. In a similar way, you can make water in a swimming pool leap straight up in the air, despite the fact that gravity prevents water from rising spontaneously. Just jump in the pool. The energy you add going in more than compensates for the force of gravity holding the water back. When the splitting of ATP molecules drives an energy-requiring reaction in a cell, the two parts of the reaction, ATP splitting and endergonic, take place in concert. In some cases, the two parts both occur on the surface of the same enzyme. They are physically linked, or coupled, like two legs walking. In other cases, a high-energy phosphate from ATP attaches to the protein catalyzing the endergonic process, activating it, figure 9.4. Coupling energy-requiring reactions to the splitting of ATP in this way is one of the key tools cells use to manage energy. Synopsis. The catabolism of glucose into carbon dioxide and water in living organisms releases about 720 kilocalories, 3012 kJ, of energy per mole of glucose. This energy is captured in ATP, which stores the energy by linking charged phosphate groups near one another. When the phosphate bonds in ATP are hydrolyzed, energy is released and available to do work. Section 9.2. Cellular respiration oxidizes food molecules. An overview of glucose catabolism. Cells are able to make ATP from the catabolism of organic molecules in two different ways. 1. Substrate-level phosphorylation. In the first, called substrate-level phosphorylation, ATP is formed by transferring a phosphate group directly to ADP from a phosphate-bearing intermediate, figure 9.5. During glycolysis, discussed below, the chemical bonds of glucose are shifted around in reactions that provide the energy required to form ATP. 2. Aerobic respiration. In the second, called aerobic respiration, ATP forms as electrons are harvested, transferred along the electron transport chain, and eventually donated to oxygen gas. Eukaryotes produce the majority of their ATP from glucose in this way. In most organisms, these two processes are combined. To harvest energy to make ATP from the sugar glucose in the presence of oxygen, the cell carries out a complex series of enzyme-catalyzed reactions that occur in 4 stages. The first stage captures energy by substrate-level phosphorylation through glycolysis. The following 3 stages carry out aerobic respiration by oxidizing the end product of glycolysis. Glycolysis. Stage 1, Glycolysis. The first stage of extracting energy from glucose is a 10-reaction biochemical pathway called glycolysis that produces ATP by substrate-level phosphorylation. The enzymes that catalyze the glycolytic reactions are in the cytoplasm of the cell, not bound to any membrane or organelle. 2 ATP molecules are used up early in the pathway, and 4 ATP molecules are formed by substrate-level phosphorylation. This yields a net of 2 ATP molecules for each molecule of glucose catabolized. In addition, 4 electrons are harvested as NADH that can be used to form ATP by aerobic respiration. Still, the total yield of ATP is small. When the glycolytic process is completed, the 2 molecules of pyruvate that are formed still contain most of the energy the original glucose molecule held. Aerobic respiration. Stage 2, Pyruvate oxidation. In the second stage, pyruvate, the end product from glycolysis, is converted into carbon dioxide and a 2-carbon molecule called acetyl-CoA. For each molecule of pyruvate converted, 1 molecule of NAD+ is reduced to NADH. Stage 3, the Krebs cycle. The third stage introduces this acetyl-CoA into a cycle of 9 reactions called the Krebs cycle, named after the British biochemist, Sir Hans Krebs, who discovered it. The Krebs cycle is also called the citric acid cycle, for the citric acid, or citrate, formed in its first step, and less commonly, the tricarboxylic acid cycle, because citrate has 3 carboxyl groups. In the Krebs cycle, 2 more ATP molecules are extracted by substrate-level phosphorylation, and a large number of electrons are removed by the reduction of NAD+ to NADH. Stage 4, Electron Transport Chain. In the fourth stage, the energetic electrons carried by NADH are employed to drive the synthesis of a large amount of ATP by the electron transport chain. Pyruvate oxidation, the reactions of the Krebs cycle, and ATP production by electron transport chains occur within many forms of bacteria and inside the mitochondria of all eukaryotes. Recall from Chapter 5 that mitochondria are thought to have evolved from bacteria. Although plants and algae can produce ATP by photosynthesis, they also produce ATP by aerobic respiration, just as animals and other non-photosynthetic eukaryotes do. Figure 9.6 provides an overview of aerobic respiration. Anaerobic respiration. In the presence of oxygen, cells can respire aerobically, using oxygen to accept the electrons harvested from food molecules. In the absence of oxygen to accept the electrons, some organisms can still respire anaerobically, using inorganic molecules to accept the electrons. For example, many bacteria use sulfur, nitrate, or other inorganic compounds as the electron acceptor in place of oxygen. Methanogens. Among the heterotrophs that practice anaerobic respiration are primitive archaebacteria such as the thermophiles discussed in Chapter 4. Some of these, called methanogens, use CO2 as the electron acceptor, reducing CO2 to CH4, methane, with the hydrogens derived from organic molecules produced by other organisms. Sulfur bacteria. Evidence of a second anaerobic respiratory process among primitive bacteria is seen in a group of rocks about 2.7 billion years old, known as the Woman River Iron Formation. Organic material in these rocks is enriched for the light isotope of sulfur, 32S, relative to the heavier isotope 34S. No known geochemical process produces such enrichment, but biological sulfur reduction does, in a process still carried out today by certain primitive bacteria. In this sulfate respiration, the bacteria derive energy from the reduction of inorganic sulfates, SO4, to H2S. The hydrogen atoms are obtained from organic molecules other organisms produce. These bacteria thus do the same thing methanogens do, but they use SO4 as the oxidizing, that is, electron-accepting, agent in place of CO2. The sulfate reducers set the stage for the evolution of photosynthesis, creating an environment rich in H2S. As discussed in Chapter 8, the first form of photosynthesis obtained hydrogens from H2S using the energy of sunlight. Synopsis. In aerobic respiration, the cell harvests energy from glucose molecules in a sequence of 4 major pathways: glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain. Oxygen is the final electron acceptor. Anaerobic respiration donates the harvested electrons to other inorganic compounds. Stage 1, Glycolysis. The metabolism of primitive organisms focused on glucose. Glucose molecules can be dismantled in many ways, but primitive organisms evolved a glucose catabolizing process that releases enough free energy to drive the synthesis of ATP in coupled reactions. This process, called glycolysis, occurs in the cytoplasm and involves a sequence of 10 reactions that convert glucose into 2 3-carbon molecules of pyruvate, figure 9.7. For each molecule of glucose that passes through this transformation, the cell nets 2 ATP molecules by substrate-level phosphorylation. Priming. The first half of glycolysis consists of 5 sequential reactions that convert 1 molecule of glucose into 2 molecules of the 3-carbon compound, glyceraldehyde 3-phosphate, G3P. These reactions demand the expenditure of ATP, so they are an energy-requiring process. Step A, glucose priming. 3 reactions, prime glucose by changing it into a compound that can be cleaved readily into 2 3-carbon phosphorylated molecules. 2 of these reactions require the cleavage of ATP, so this step requires the cell to use 2 ATP molecules. Step B, cleavage and rearrangement. In the first of the remaining pair of reactions, the 6-carbon product of step A is split into 2 3-carbon molecules. One is G3P, and the other is then converted to G3P by the second reaction, figure 9.8. Substrate-level phosphorylation. In the second half of glycolysis, 5 more reactions convert G3P into pyruvate in an energy-yielding process that generates ATP. Overall, then, glycolysis is a series of 10 enzyme-catalyzed reactions in which some ATP is invested in order to produce more. Step C, oxidation. 2 electrons and 1 proton are transferred from G3P to NAD+, forming NADH. Note that NAD+ is an ion, and that both electrons in the new covalent bond come from G3P. Step D, ATP generation. 4 reactions convert G3P into another 3-carbon molecule, pyruvate. This process generates 2 ATP molecules, see figure 9.5. Because each glucose molecule is split into 2 G3P molecules, the overall reaction sequence yields 2 molecules of ATP, as well as 2 molecules of NADH and 2 of pyruvate. For ATP, 2 ATP for each of the 2 G3P molecules in step D. Minus 2 ATP, used in the 2 reactions in step A. Leaves 2 molecules of ATP. Under the non-standard conditions within a cell, each ATP molecule produced represents the capture of about 12 kilocalories, 50 kilojoules, of energy per mole of glucose, rather than the 7.3 traditionally quoted for standard conditions. This means glycolysis harvests about 24 kilocalories per mole, 100 kilojoules per mole. This is not a great deal of energy. The total energy content of the chemical bonds of glucose is 686 kilocalories, 2,870 kilojoules, per mole, so glycolysis harvests only 3.5% of the chemical energy of glucose. Although far from ideal in terms of the amount of energy it releases, glycolysis does generate ATP. For more than 1 billion years during the anaerobic first stages of life on Earth, it was the primary way heterotrophic organisms generated ATP from organic molecules. Like many biochemical pathways, glycolysis is believed to have evolved backward, with the last steps in the process being the most ancient. Thus, the second half of glycolysis, the ATP-yielding breakdown of G3P, may have been the original process early heterotrophs used to generate ATP. The synthesis of G3P from glucose would have appeared later, perhaps when alternative sources of G3P were depleted. All cells use glycolysis. The glycolytic reaction sequence is thought to have been among the earliest of all biochemical processes to evolve. It uses no molecular oxygen and occurs readily in an anaerobic environment. All of its reactions occur free in the cytoplasm, none is associated with any organelle or membrane structure. Every living creature is capable of carrying out glycolysis. Most present-day organisms, however, can extract considerably more energy from glucose through aerobic respiration. Why does glycolysis take place even now, since its energy yield in the absence of oxygen is comparatively so paltry? The answer is that evolution is an incremental process. Change occurs by improving on past successes. In catabolic metabolism, glycolysis satisfied the one essential evolutionary criterion: it was an improvement. Cells that could not carry out glycolysis were at a competitive disadvantage, and only cells capable of glycolysis survived the early competition of life. Later improvements in catabolic metabolism built on this success. Glycolysis was not discarded during the course of evolution, rather, it served as the starting point for the further extraction of chemical energy. Metabolism evolved as one layer of reactions added to another, just as successive layers of paint cover the walls of an old building. Nearly every present-day organism carries out glycolysis as a metabolic memory of its evolutionary past. Closing the metabolic circle, the regeneration of NAD+. Inspect for a moment the net reaction of the glycolytic sequence. Glucose plus 2 ADP plus 2 phosphate plus 2 NAD plus produces 2 pyruvate plus 2 ATP plus 2 NADH plus 2 H plus 2 H2O. You can see that 3 changes occur in glycolysis. 1. Glucose is converted into 2 molecules of pyruvate. 2. 2 molecules of ADP are converted into ATP via substrate-level phosphorylation. And 3. 2 molecules of NAD+ are reduced to NADH. The need to recycle NADH. As long as food molecules that can be converted into glucose are available, a cell can continually churn out ATP to drive its activities. In doing so, however, it accumulates NADH and depletes the pool of NAD+ molecules. A cell does not contain a large amount of NAD+, and for glycolysis to continue, NADH must be recycled into NAD+. Some other molecule than NAD+ must ultimately accept the hydrogen atom taken from G3P and be reduced. 2 molecules can carry out this key task, figure 9.9. 1. Aerobic respiration. Oxygen is an excellent electron acceptor. Through a series of electron transfers, the hydrogen atom taken from G3P can be donated to oxygen, forming water. This is what happens in the cells of eukaryotes in the presence of oxygen. Because air is rich in oxygen, this process is also referred to as aerobic metabolism. 2. Fermentation. When oxygen is unavailable, an organic molecule can accept the hydrogen atom instead. Such fermentation plays an important role in the metabolism of most organisms, figure 9.10, even those capable of aerobic respiration. The fate of the pyruvate that is produced by glycolysis depends upon which of these 2 processes takes place. The aerobic respiration path starts with the oxidation of pyruvate to a molecule called acetyl-CoA, which is then further oxidized in a series of reactions called the Krebs cycle. The fermentation path, by contrast, involves the reduction of all or part of pyruvate. We will start by examining aerobic respiration, then look briefly at fermentation. Synopsis. Glycolysis generates a small amount of ATP by reshuffling the bonds of glucose molecules. In glycolysis, 2 molecules of NAD+ are reduced to NADH. NAD+ must be regenerated for glycolysis to continue unabated. Stage 2, the oxidation of pyruvate. In the presence of oxygen, the oxidation of glucose that begins in glycolysis continues where glycolysis leaves off, with pyruvate. In eukaryotic organisms, the extraction of additional energy from pyruvate takes place exclusively inside mitochondria. The cell harvests pyruvate's considerable energy in 2 steps, first, by oxidizing pyruvate to form acetyl-CoA, and then by oxidizing acetyl-CoA in the Krebs cycle. Producing acetyl-CoA. Pyruvate is oxidized in a single decarboxylation reaction that cleaves off one of pyruvate's 3 carbons. This carbon then departs as CO2, figure 9.11, top. This reaction produces a 2-carbon fragment called an acetyl group, as well as a pair of electrons and their associated hydrogen, which reduce NAD+ to NADH. The reaction is complex, involving 3 intermediate stages, and is catalyzed within mitochondria by a multi-enzyme complex. As Chapter 8 noted, such a complex organizes a series of enzymatic steps so that the chemical intermediates do not diffuse away or undergo other reactions. Within the complex, component polypeptides pass the substrates from one enzyme to the next, without releasing them. Pyruvate dehydrogenase, the complex of enzymes that removes CO2 from pyruvate, is one of the largest enzymes known. It contains 60 subunits. In the course of the reaction, the acetyl group removed from pyruvate combines with a cofactor called coenzyme A, CoA, forming a compound known as acetyl-CoA. Pyruvate plus NAD plus CoA produces acetyl-CoA plus NADH plus CO2. This reaction produces a molecule of NADH, which is later used to produce ATP. Of far greater significance than the reduction of NAD+ to NADH, however, is the production of acetyl-CoA, figure 9.11, bottom. Acetyl-CoA is important because so many different metabolic processes generate it. Not only does the oxidation of pyruvate, an intermediate in carbohydrate catabolism, produce it, but the metabolic breakdown of proteins, fats, and other lipids also generate acetyl-CoA. Indeed, almost all molecules catabolized for energy are converted into acetyl-CoA. Acetyl-CoA is then channeled into fat synthesis or into ATP production, depending on the organism's energy requirements. Acetyl-CoA is a key point of focus for the many catabolic processes of the eukaryotic cell. Using acetyl-CoA. Although the cell forms acetyl-CoA in many ways, only a limited number of processes use acetyl-CoA. Most of it is either directed toward energy storage, lipid synthesis, for example, or oxidized in the Krebs cycle to produce ATP. Which of these two options is taken depends on the level of ATP in the cell. When ATP levels are high, the oxidative pathway is inhibited, and acetyl-CoA is channeled into fatty acid synthesis. This explains why many animals, humans included, develop fat reserves when they consume more food than their bodies require. Alternatively, when ATP levels are low, the oxidative pathway is stimulated, and acetyl-CoA flows into energy-producing oxidative metabolism. Synopsis. In the second energy-harvesting stage of glucose catabolism, pyruvate is decarboxylated, yielding acetyl-CoA, NADH, and CO2. This process occurs within the mitochondrion. Stage 3, the Krebs cycle. After glycolysis catabolizes glucose to produce pyruvate, and pyruvate is oxidized to form acetyl-CoA, the third stage of extracting energy from glucose begins. In this third stage, acetyl-CoA is oxidized in a series of 9 reactions called the Krebs cycle. These reactions occur in the matrix of mitochondria. In this cycle, the 2-carbon acetyl group of acetyl-CoA combines with a 4-carbon molecule called oxaloacetate, figure 9.12. The resulting 6-carbon molecule then goes through a sequence of electron-yielding oxidation reactions, during which 2 CO2 molecules split off, restoring oxaloacetate. The oxaloacetate is then recycled to bind to another acetyl group. In each turn of the cycle, a new acetyl group replaces the 2 CO2 molecules lost, and more electrons are extracted to drive proton pumps that generate ATP. Overview of the Krebs cycle. The 9 reactions of the Krebs cycle occur in 2 steps. Step A, priming. 3 reactions prepare the 6-carbon molecule for energy extraction. First, acetyl-CoA joins the cycle, and then chemical groups are rearranged. Step B, energy extraction. 4 of the 6 reactions in this step are oxidations in which electrons are removed, and 1 generates an ATP equivalent directly by substrate-level phosphorylation. The reactions of the Krebs cycle. The Krebs cycle consists of 9 sequential reactions that cells use to extract energetic electrons and drive the synthesis of ATP, figure 9.13. A 2-carbon group from acetyl-CoA enters the cycle at the beginning, and 2 CO2 molecules and several electrons are given off during the cycle. Reaction 1, condensation. The 2-carbon group from acetyl-CoA joins with a 4-carbon molecule, oxaloacetate, to form a 6-carbon molecule, citrate. This condensation reaction is irreversible, committing the 2-carbon acetyl group to the Krebs cycle. The reaction is inhibited when the cell's ATP concentration is high and stimulated when it is low. Hence, when the cell possesses ample amounts of ATP, the Krebs cycle shuts down and acetyl-CoA is channeled into fat synthesis. Reactions 2 and 3, isomerization. Before the oxidation reactions can begin, the hydroxyl, OH, group of citrate must be repositioned. This is done in 2 steps. First, a water molecule is removed from one carbon, then, water is added to a different carbon. As a result, an H group and an OH group change positions. The product is an isomer of citrate called isocitrate. Reaction 4, the first oxidation. In the first energy-yielding step of the cycle, isocitrate undergoes an oxidative decarboxylation reaction. First, isocitrate is oxidized, yielding a pair of electrons that reduce a molecule of NAD+ to NADH. Then the oxidized intermediate is decarboxylated, the central carbon atom splits off to form CO2, yielding a 5-carbon molecule called alpha-ketoglutarate. Reaction 5, the second oxidation. Next, alpha-ketoglutarate is decarboxylated by a multi-enzyme complex similar to pyruvate dehydrogenase. The succinyl group left after the removal of CO2 joins to coenzyme A, forming succinyl-CoA. In the process, 2 electrons are extracted, and they reduce another molecule of NAD+ to NADH. Reaction 6, substrate-level phosphorylation. The linkage between the 4-carbon succinyl group and CoA is a high-energy bond. In a coupled reaction similar to those that take place in glycolysis, this bond is cleaved, and the energy released drives the phosphorylation of guanosine diphosphate, GDP, forming guanosine triphosphate, GTP. GTP is readily converted into ATP, and the 4-carbon fragment that remains is called succinate. Reaction 7, the third oxidation. Next, succinate is oxidized to fumarate. The free energy change in this reaction is not large enough to reduce NAD+. Instead, flavin adenine dinucleotide (FAD) is the electron acceptor. Unlike NAD+, FAD is not free to diffuse within the mitochondrion, it is an integral part of the inner mitochondrial membrane. Its reduced form, FADH2, contributes electrons to the electron transport chain in the membrane. Reactions 8 and 9, regeneration of oxaloacetate. In the final 2 reactions of the cycle, a water molecule is added to fumarate, forming malate. Malate is then oxidized, yielding a 4-carbon molecule of oxaloacetate and 2 electrons that reduce a molecule of NAD+ to NADH. Oxaloacetate, the molecule that began the cycle, is now free to combine with another 2-carbon acetyl group from acetyl-CoA and reinitiate the cycle. The products of the Krebs cycle. In the process of aerobic respiration, glucose is entirely consumed. The 6-carbon glucose molecule is first cleaved into a pair of 3-carbon pyruvate molecules during glycolysis. One of the carbons of each pyruvate is then lost as CO2 in the conversion of pyruvate to acetyl-CoA. 2 other carbons are lost as CO2 during the oxidations of the Krebs cycle. All that is left to mark the passing of the glucose molecule into 6 CO2 molecules is its energy, some of which is preserved in 4 ATP molecules and in the reduced state of 12 electron carriers. 10 of these carriers are NADH molecules, the other 2 are FADH2. Synopsis. The Krebs cycle generates 2 ATP molecules per molecule of glucose, the same number generated by glycolysis. More importantly, the Krebs cycle and the oxidation of pyruvate harvest many energized electrons, which can be directed to the electron transport chain to drive the synthesis of much more ATP. Harvesting energy by extracting electrons. To understand how cells direct some of the energy released during glucose catabolism into ATP production, we need to take a closer look at the electrons in the CH bonds of the glucose molecule. We stated in Chapter 8 that when an electron is removed from one atom and donated to another, the electron's potential energy of position is also transferred. In this process, the atom that receives the electron is reduced. We spoke of reduction in an all-or-none fashion, as if it involved the complete transfer of an electron from one atom to another. Often this is just what happens. However, sometimes a reduction simply changes the degree of sharing within a covalent bond. Let us now revisit that discussion and consider what happens when the transfer of electrons is incomplete. A closer look at oxidation reduction. The catabolism of glucose is an oxidation-reduction reaction. The covalent electrons in the CH bonds of glucose are shared approximately equally between the C and H atoms because carbon and hydrogen nuclei have about the same affinity for valence electrons, that is, they exhibit similar electronegativity. However, when the carbon atoms of glucose react with oxygen to form carbon dioxide, the electrons in the new covalent bonds take a different position. Instead of being shared equally, the electrons that were associated with the carbon atoms in glucose shift far toward the oxygen atom in CO2 because oxygen is very electronegative. Since these electrons are pulled farther from the carbon atoms of glucose have been oxidized, loss of electrons, and the oxygen atoms reduced, gain of electrons. Similarly, when the hydrogen atoms of glucose combine with oxygen atoms to form water, the oxygen atoms draw the shared electrons strongly toward them. Again, oxygen is reduced and glucose is oxidized. In this reaction, oxygen is an oxidizing, electron-attracting, agent because it oxidizes the atoms of glucose. Releasing energy. The key to understanding the oxidation of glucose is to focus on the energy of the shared electrons. In a covalent bond, energy must be added to remove an electron from an atom, just as energy must be used to roll a boulder up a hill. The more electronegative the atom, the steeper the energy hill that must be climbed to pull an electron away from it. However, energy is released when an electron is shifted away from a less electronegative atom and closer to a more electronegative atom, just as energy is released when a boulder is allowed to roll down a hill. In the catabolism of glucose, energy is released when glucose is oxidized, as electrons relocate closer to oxygen, figure 9.14. Glucose is an energy-rich food because it has an abundance of CH bonds. Viewed in terms of oxidation reduction, glucose possesses a wealth of electrons held far from their atoms, all with the potential to move closer toward oxygen. In oxidative respiration, energy is released not simply because the hydrogen atoms of the CH bonds are transferred from glucose to oxygen, but because the positions of the valence electrons shift. This shift releases energy that can be used to make ATP. Harvesting the energy in stages. It is generally true that the larger the release of energy in any single step, the more of that energy is released as heat, random molecular motion, and the less there is available to be channeled into more useful paths. In the combustion of gasoline, the same amount of energy is released whether all of the gasoline in a car's gas tank explodes at once, or whether the gasoline burns in a series of very small explosions inside the cylinders. By releasing the energy in gasoline a little at a time, the harvesting efficiency is greater and more of the energy can be used to push the pistons and move the car. The same principle applies to the oxidation of glucose inside a cell. If all of the hydrogens were transferred to oxygen in one explosive step, releasing all of the free energy at once, the cell would recover very little of that energy in a useful form. Instead, cells burn their fuel much as a car does, a little at a time. The 6 hydrogens in the CH bonds of glucose are stripped off in stages in the series of enzyme-catalyzed reactions collectively referred to as glycolysis and the Krebs cycle. We have had a great deal to say about these reactions already in this chapter. Recall that the hydrogens are removed by transferring them to a coenzyme carrier, NAD+, figure 9.15. Discussed in Chapter 8, NAD+ is a very versatile electron acceptor, shuttling energy-bearing electrons throughout the cell. In harvesting the energy of glucose, NAD+ acts as the primary electron acceptor. Following the electrons. As you examine these reactions, try not to become confused by the changes in electrical charge. Always follow the electrons. Enzymes extract 2 hydrogens, that is, 2 electrons and 2 protons, from glucose and transfer both electrons and one of the protons to NAD+. The other proton is released as a hydrogen ion, H+, into the surrounding solution. This transfer converts NAD+ into NADH, that is, 2 negative electrons and 1 positive proton are added to 1 positively charged NAD+ to form NADH, which is electrically neutral. Energy captured by NADH is not harvested all at once. Instead of being transferred directly to oxygen, the two electrons carried by NADH are passed along the electron transport chain if oxygen is present. This chain consists of a series of molecules, mostly proteins, embedded within the inner membranes of mitochondria. NADH delivers electrons to the top of the electron transport chain and oxygen captures them at the bottom. The oxygen then joins with hydrogen ions to form water. At each step in the chain, the electrons move to a slightly more electronegative carrier, and their positions shift slightly. Thus, the electrons move down an energy gradient. The entire process releases a total of 53 kilocalories per mole, 222 kilojoules per mole, under standard conditions. The transfer of electrons along this chain allows the energy to be extracted gradually. In the next section, we will discuss how this energy is put to work to drive the production of ATP. Synopsis. The catabolism of glucose involves a series of oxidation-reduction reactions that release energy by repositioning electrons closer to oxygen atoms. Energy is thus harvested from glucose molecules in gradual steps, using NAD+ as an electron carrier. Stage 4, the electron transport chain. The NADH and FADH2 molecules formed during the first three stages of aerobic respiration each contain a pair of electrons that were gained when NAD+ and FAD were reduced. The NADH molecules carry their electrons to the inner mitochondrial membrane, where they transfer the electrons to a series of membrane-associated proteins collectively called the electron transport chain. Moving electrons through the electron transport chain. The first of the proteins to receive the electrons is a complex, membrane-embedded enzyme called NADH dehydrogenase. A carrier called ubiquinone then passes the electrons to a protein cytochrome complex called the BC1 complex. This complex, along with others in the chain, operates as a proton pump, driving a proton out across the membrane. Cytochromes are respiratory proteins that contain heme groups, complex carbon rings with many alternating single and double bonds and an iron atom in the center. The electron is then carried by another carrier, cytochrome C, to the cytochrome oxidase complex. This complex uses for such electrons to reduce a molecule of oxygen. Each oxygen then combines with two hydrogen ions to form water. O2 plus 4H plus 4E minus produces 2H2O. This series of membrane-associated electron carriers is collectively called the electron transport chain, figure 9.16. NADH contributes its electrons to the first protein of the electron transport chain, NADH dehydrogenase. FADH2, which is always attached to the inner mitochondrial membrane, feeds its electrons into the electron transport chain later, to ubiquinone. It is the availability of a plentiful electron acceptor, often oxygen, that makes oxidative respiration possible. As we'll see in Chapter 10, the electron transport chain used in aerobic respiration is similar to, and may well have evolved from, the chain employed in aerobic photosynthesis. Synopsis. The electron transport chain is a series of 5 membrane-associated proteins. Electrons delivered by NADH and FADH2 are passed from protein to protein along the chain, like a baton in a relay race. Building an electrochemical gradient. In eukaryotes, aerobic metabolism takes place within the mitochondria present in virtually all cells. The internal compartment, or matrix, of a mitochondrion contains the enzymes that carry out the reactions of the Krebs cycle. As the electrons harvested by oxidative respiration are passed along the electron transport chain, the energy they release transports protons out of the matrix and into the outer compartment, sometimes called the intermembrane space. 3 transmembrane proteins in the inner mitochondrial membrane, see figure 9.16, actually accomplish the transport. The flow of excited electrons induces a change in the shape of these pump proteins, which causes them to transport protons across the membrane. The electrons contributed by NADH activate all 3 of these proton pumps, while those contributed by FADH2 activate only 2. Producing ATP, chemiosmosis. As the proton concentration in the intermembrane space rises above that in the matrix becomes slightly negative in charge. This internal negativity attracts the positively charged protons and induces them to re-enter the matrix. The higher outer concentration tends to drive protons back in by diffusion. Since membranes are relatively impermeable to ions, most of the protons that re-enter the matrix pass through special proton channels in the inner mitochondrial membrane. When the protons pass through, these channels synthesize ATP from ADP plus phosphate within the matrix. The ATP is then transported by facilitated diffusion out of the mitochondrion and into the cell's cytoplasm. Because the chemical formation of ATP is driven by a diffusion force similar to osmosis, this process is referred to as chemiosmosis, figure 9.17. Thus, the electron transport chain uses electrons harvested in aerobic respiration to pump a large number of protons across the inner mitochondrial membrane. Their subsequent re-entry into the mitochondrial matrix drives the synthesis of ATP by chemiosmosis. Figure 9.18 summarizes the overall process. Synopsis. The electrons harvested from glucose are pumped out of the mitochondrial matrix by the electron transport chain. The return of the protons into the matrix generates ATP. Summarizing aerobic respiration. How much metabolic energy does a cell actually gain from the electrons harvested from a molecule of glucose, using the electron transport chain to produce ATP by chemiosmosis? Theoretical yield. The chemiosmotic model suggests that one ATP molecule is generated for each proton pump activated by the electron transport chain. Since the electrons from NADH activate 3 pumps and those from FADH2 activate 2, we would expect each molecule of NADH and FADH2 to generate 3 and 2 ATP molecules, respectively. However, because eukaryotic cells carry out glycolysis in their cytoplasm and the Krebs cycle within their mitochondria, they must transport the 2 molecules of NADH produced during glycolysis across the mitochondrial membranes, which requires 1 ATP per molecule of NADH. Thus, the net ATP production is decreased by 2. Therefore, the overall ATP production resulting from aerobic respiration theoretically should be 4, from substrate-level phosphorylation during glycolysis, plus 30, 3 from each of 10 molecules of NADH, plus 4, 2 from each of 2 molecules of FADH2, minus 2, for transport of glycolytic NADH, equals 36 molecules of ATP, figure 9.19. Actual yield. The amount of ATP actually produced in a eukaryotic cell during aerobic respiration is somewhat lower than 36, for 2 reasons. First, the inner mitochondrial membrane is somewhat leaky to protons, allowing some of them to re-enter the matrix without passing through ATP-generating channels. Second, mitochondria often use the proton gradient generated by chemiosmosis for purposes other than ATP synthesis, such as transporting pyruvate into the matrix. Consequently, the actual measured values of ATP generated by NADH and FADH2 are closer to 2.5 for each NADH and 1.5 for each FADH2. With these corrections, the overall harvest of ATP from a molecule of glucose in a eukaryotic cell is closer to 4, from substrate-level phosphorylation, plus 25, 2.5 from each of 10 molecules of NADH, plus 3, 1.5 from each of 2 molecules of FADH2, minus 2, transport of glycolytic NADH, equals 30 molecules of ATP. The catabolism of glucose by aerobic respiration, in contrast to glycolysis, is quite efficient. Aerobic respiration in a eukaryotic cell harvests about 7.3 by 30 divided by 686 equals 32% of the energy available in glucose. By comparison, a typical car converts only about 25% of the energy in gasoline into useful energy. The efficiency of oxidative respiration at harvesting energy establishes a natural limit on the maximum length of food chains. The high efficiency of aerobic respiration was one of the key factors that fostered the evolution of heterotrophs. With this mechanism for producing ATP, it became feasible for non-photosynthetic organisms to derive metabolic energy exclusively from the oxidative breakdown of other organisms. As long as some organisms captured energy by photosynthesis, others could exist solely by feeding on them. Synopsis. Oxidative respiration produces approximately 30 molecules of ATP from each molecule of glucose in eukaryotic cells. This represents more than half of the energy in the chemical bonds of glucose. Regulating aerobic respiration. When cells possess plentiful amounts of ATP, the key reactions of glycolysis, the Krebs cycle, and fatty acid breakdown are inhibited, slowing ATP production. The regulation of these biochemical pathways by the level of ATP is an example of feedback inhibition. Conversely, when ATP levels in the cell are low, ADP levels are high, and ADP activates enzymes in the pathways of carbohydrate catabolism to stimulate the production of more ATP. Control of glucose catabolism occurs at two key points of the catabolic pathway, figure 9.20. The control point in glycolysis is the enzyme phosphofructokinase, which catalyzes reaction 3, the conversion of fructose phosphate to fructose bisphosphate. This is the first reaction of glycolysis that is not readily reversible, committing the substrate to the glycolytic sequence. High levels of ADP relative to ATP, implying a need to convert more ADP to ATP, stimulate phosphofructokinase, committing more sugar to the catabolic pathway, so do low levels of citrate, implying the Krebs cycle is not running at full tilt and needs more input. The main control point in the oxidation of pyruvate occurs at the committing step in the Krebs cycle with the enzyme pyruvate decarboxylase. It is inhibited by high levels of NADH, implying no more is needed. Another control point in the Krebs cycle is the enzyme citrate synthetase, which catalyzes the first reaction, the conversion of oxaloacetate and acetyl-CoA into citrate. High levels of ATP inhibit citrate synthetase, as well as pyruvate decarboxylase and two other Krebs cycle enzymes, shutting down the catabolic pathway. Synopsis. Relative levels of ADP and ATP regulate the catabolism of glucose at key committing reactions. A vocabulary of ATP generation. Aerobic respiration. The portion of cellular respiration that requires oxygen as an electron acceptor, it includes pyruvate oxidation, the Krebs cycle, and the electron transport chain. Anaerobic respiration. Cellular respiration in which inorganic electron acceptors other than oxygen are used, it includes glycolysis. Cellular respiration. The oxidation of organic molecules to produce ATP in which the final electron acceptor is organic, it includes aerobic and anaerobic respiration. Chemiosmosis. The passage of high-energy electrons along the electron transport chain, which is coupled to the pumping of protons across a membrane and the return of protons to the original side of the membrane through ATP-generating channels. Fermentation. Alternative ATP-producing pathway performed by some cells in the absence of oxygen, in which the final electron acceptor is an organic molecule. Maximum efficiency. The maximum number of ATP molecules generated by oxidizing a substance, relative to the free energy of that substance. In organisms, the actual efficiency is usually less than the maximum. Oxidation. The loss of an electron. In cellular respiration, high-energy electrons are stripped from food molecules, oxidizing them. Photosynthesis. The chemiosmotic generation of ATP and complex organic molecules powered by the energy derived from light. Substrate-level phosphorylation. The generation of ATP by the direct transfer of a phosphate group to ADP from another phosphorylated molecule. Section 9.3. Catabolism of proteins and fats can yield considerable energy. Glucose is not the only source of energy. Thus far we have discussed oxidative respiration of glucose, which organisms obtain from the digestion of carbohydrates or from photosynthesis. Other organic molecules than glucose, particularly proteins and fats, are also important sources of energy, figure 9.21. Cellular respiration of protein. Proteins are first broken down into their individual amino acids. The nitrogen-containing side group, the amino group, is then removed from each amino acid in a process called deamination. A series of reactions convert the carbon chain that remains into a molecule that takes part in glycolysis or the Krebs cycle. For example, alanine is converted into pyruvate, glutamate into alpha-ketoglutarate, figure 9.22, and aspartate into oxaloacetate. The reactions of glycolysis and the Krebs cycle then extract the high-energy electrons from these molecules and put them to work making ATP. Cellular respiration of fat. Fats are broken down into fatty acids plus glycerol. The tails of fatty acids typically have 16 or more CH2 links, and the many hydrogen atoms in these long tails provide a rich harvest of energy. Fatty acids are oxidized in the matrix of the mitochondrion. Enzymes there remove the 2-carbon acetyl groups from the end of each fatty acid tail until the entire fatty acid is converted into acetyl groups, figure 9.23. Each acetyl group then combines with coenzyme A to form acetyl-CoA. This process is known as beta-oxidation. How much ATP does the catabolism of fatty acids produce? Let's compare a hypothetical 6-carbon fatty acid with the 6-carbon glucose molecule, which we've said yields about 30 molecules of ATP in a eukaryotic cell. 2 rounds of beta-oxidation would convert the fatty acid into 3 molecules of acetyl-CoA. Each round requires 1 molecule of ATP to prime the process, but it also produces 1 molecule of NADH and 1 of FADH2. These molecules together yield 4 molecules of ATP, assuming 2.5 ATPs per NADH and 1.5 ATPs per FADH2. The oxidation of each acetyl-CoA in the Krebs cycle ultimately produces an additional 10 molecules of ATP. Overall, then, the ATP yield of a 6-carbon fatty acid would be approximately 8, from 2 rounds of beta-oxidation, minus 2, for priming those 2 rounds, plus 30, from oxidizing the 3 acetyl-CoAs, equals 36 molecules of ATP. Therefore, the respiration of a 6-carbon fatty acid yields 20% more ATP than the respiration of glucose. Moreover, a fatty acid of that size would weigh less than 2/3 as much as glucose, so a gram of fatty acid contains more than twice as many kilocalories as a gram of glucose. That is why fat is a storage molecule for excess energy in many types of animals. If excess energy were stored instead as carbohydrate, as it is in plants, animal bodies would be much bulkier. Synopsis. Proteins, fats, and other organic molecules are also metabolized for energy. The amino acids of proteins are first deaminated, while fats undergo a process called beta-oxidation. Metabolic efficiency and the length of food chains. It has been estimated that a heterotroph limited to glycolysis captures only 3.5% of the energy in the food it consumes. Hence, if such a heterotroph preserves 3.5% of the energy in the autotrophs it consumes, then any other heterotrophs that consume the first heterotroph will capture through glycolysis 3.5% of the energy in it, or 0.12% of the energy available in the original autotrophs. A very large base of autotrophs would thus be needed to support a small number of heterotrophs. When organisms became able to extract energy from organic molecules by oxidative metabolism, this constraint became far less severe, because the efficiency of oxidative respiration is estimated to be about 52 to 63%. This increased efficiency results in the transmission of much more energy from one trophic level to another than does glycolysis. A trophic level is a step in the movement of energy through an ecosystem. The efficiency of oxidative metabolism has made possible the evolution of food chains, in which autotrophs are consumed by heterotrophs, which are consumed by other heterotrophs, and so on. You will read more about food chains in Chapter 28. Even with oxidative metabolism, approximately 2/3 of the available energy is lost at each trophic level, and that puts a limit on how long a food chain can be. Most food chains, like the one illustrated in Figure 9.a, involve only 3 or rarely 4 trophic levels. Too much energy is lost at each transfer to allow chains to be much longer than that. For example, it would be impossible for a large human population to subsist by eating lions captured from the Serengeti plain of Africa, the amount of grass available there would not support enough zebras and other herbivores to maintain the number of lions needed to feed the human population. Thus, the ecological complexity of our world is fixed in a fundamental way by the chemistry of oxidative respiration. Section 9.4. Cells can metabolize food without oxygen. Fermentation. In the absence of oxygen, aerobic metabolism cannot occur, and cells must rely exclusively on glycolysis to produce ATP. Under these conditions, the hydrogen atoms generated by glycolysis are donated to organic molecules in a process called fermentation. Bacteria carry out more than a dozen kinds of fermentations, all using some form of organic molecule to accept the hydrogen atom from NADH and thus recycle. NAD+. Organic molecule plus NADH produces reduced organic molecule plus NAD+. Often the reduced organic compound is an organic acid, such as acetic acid, butyric acid, propionic acid, or lactic acid, or an alcohol. Ethanol fermentation. Eukaryotic cells are capable of only a few types of fermentation. In one type, which occurs in single-celled fungi called yeast, the molecule that accepts hydrogen from NADH is pyruvate, the end product of glycolysis itself. Yeast enzymes remove a terminal CO2 group from pyruvate through decarboxylation, producing a 2-carbon molecule called acetaldehyde. The CO2 released causes bread made with yeast to rise, while bread made without yeast, unleavened bread, does not. The acetaldehyde accepts a hydrogen atom from NADH, producing NAD+ and ethanol, ethyl alcohol. This particular type of fermentation is of great interest to humans, since it is the source of the ethanol in wine and beer, figure 9.24. Ethanol is a byproduct of fermentation that is actually toxic to yeast. As it approaches a concentration of about 12%, it begins to kill the yeast. That explains why naturally fermented wine contains only about 12% ethanol. Lactic acid fermentation. Most animal cells regenerate NAD+ without decarboxylation. Muscle cells, for example, use an enzyme called lactate dehydrogenase to transfer a hydrogen atom from NADH back to the pyruvate that is produced by glycolysis. This reaction converts pyruvate into lactic acid and regenerates NAD+ from NADH. It therefore closes the metabolic circle, allowing glycolysis to continue as long as glucose is available. Circulating blood removes excess lactate, the ionized form of lactic acid, from muscles, but when removal cannot keep pace with production, the accumulating lactic acid interferes with muscle function and contributes to muscle fatigue. Synopsis. In fermentation, which occurs in the absence of oxygen, the electrons that result from the glycolytic breakdown of glucose are donated to an organic molecule, regenerating NAD+ from NADH.